Okay, so everybody knew we were going to start Chapter 9 today. Good. 11? We already had a test on that. Okay, so what are we talking about? Muscles. Muscle and muscle tissue. One of the only complaints I have about this book is the way it's set up. Because if we talk about muscles and we don't understand what neurons does with respect to muscles, then it can be a little confusing. So that's why we did what we did as far as presenting topics in this class. The other thing is, you don't want to be cramming the nervous system in at the end of the semester, do you? No. Just think, the worst part is done. Well, of this semester. See? So this stuff's not too bad. Although some of you might not be too happy about memorizing some of the muscles because they're not really easy to pronounce. Yes? Shh, don't say anything. Now, the other thing, before I hit this topic, I want to remind you of is you have some really awesome software, don't you? Yeah, you have a PAL. You have a PAL, and the PAL will come in handy for muscles. When you go to PAL, and where do you find PAL if you don't have that cute little disc in your hot little hand? Yeah, you can get there through Mastering. Go to PAL. Does anybody know what I'm talking about or not know what I'm talking about? At this point in the semester, you should know what I'm talking about. In there, you'll see models. And there is models for muscles. And guess what? If you didn't know it already, I'm going to tell you, and I know it's going to be very exciting for you. But if you go in there, and it's similar to the histology, you press on, there's, it'll boom, bring up a model of a leg. And you know how they have labels? You can label or unlabel to study. Those of you who haven't looked at PAL, probably a good idea to do that now. But did you know that if you go press on the word, it pronounces it for you? It's awesome. Okay? Yeah, I'm a geek. Well, and I've also learned that I'm mispronouncing some of those muscles for so many years. It's like, oh, is that the way you say that? So go use that. That's good. Because you hear it, you see it. Hopefully those two things will make you know it so you can pass your test. Right? Huh? One can always hope. The other piece of wonderfulness you have for this chapter also is interactive physiology. Does everybody know what I'm talking about? Find it in the same place, over on the left-hand side. And we're going to actually, because it does a better job of explaining things. I explain it pretty well, but when I can see it as it's being explained, it makes more sense. Does that make sense to you? So if I can visualize what's going on as someone is speaking, that helps me learn things a little bit better. So the interactive physiology, the sliding filament theory, is something that I highly suggest you view several times to more than once this weekend. Because it just pulls everything together and it makes much more sense. I can come up here and wave my arms and explain it to you, but if you can see it while it's being explained, it'll make more sense. Okay? So we're going to go through that in a moment. So, muscle tissue. What is it? What's it for? Moving stuff. Like, it's moving your skeleton. What kind of muscle is that? Skeletal muscle. We know this already, right? What are moving what other stuff? Organ, like the heart. So we have cardiac muscle. And then smooth muscle moves what? Stuff through your tubes. Yes? Anybody eat lunch, breakfast, or any meal? Ever? How does that meal get from the beginning to the end of your digestive system? Smooth muscle is going to help push it through a process called peristalsis. So the types of muscle are described in the beginning of your textbook. Now, some of the different characteristics of muscles. They are excitable. Does that mean you tell them something good and they jump up and down? But they have the ability to jump up and down. That's what excitable means. So they have the ability to do what? Yep, respond. They're irritable. And no, that doesn't mean cranky. They can be cranky. They can be crampy. Right? So they can contract. They can stretch. And they have the ability to do both and be very elastic. So those are some of the special characteristics of muscle tissue. So we need them for movement. We need them so we don't fall down on our face. Posture. To allow us to move through space in a fluid manner. They're going to help stabilize our joints so we can move through the space in a fluid manner. Anybody ever get some muscles messed up, a muscle tear, a muscle pull, a sprain, a strain? Can you move through space fluidly? No. So, you can, but it hurts. So it helps to stabilize joints. Generate heat. And some of you might see an example of this next week after your Thanksgiving meal. Run the marathon before the meal. It's better for digestion. Generate heat. What's she talking about? You ready for? Yes? Awesome. It's the byproduct of muscle contraction. So one of the byproducts of muscle contraction is heat. Now, here's my example next week. After you eat your big Thanksgiving meal, some of you might curl up on the couch, take a little nappy. And everybody else around's doing work, cleaning the dishes while you're taking your nap, right? If you're lucky and you're the person that gets to lay on the couch and not the person that has to do the dishes, right? And you get up from your nap and everybody's finally finishing the dishes and you say, oh, it's cold. Can we put the heat on? And everybody else is like, it's fine in here. What's your problem? Yes? Why are you cold and they're fine? Because you were taking a nappy on the couch and not moving a lot of your what? Moving your muscles. So muscles give off heat and help to maintain body temperature. And that's one of their very important functions. What happens when you get really cold? You actually force muscle contraction. Little contractions in hair follicle muscles called erector pili muscles cause you to get what? Goosebumps. And those are just contractions of muscles to generate heat. What are some of the additional functions of muscle? They are going to help protect fragile organs, and that's what we call the viscera. And they include things like the stomach, regulate passage through tubes, like the digestive system and the small intestine and large intestine and all of that good stuff. And they allow certain amounts of light to come in and out of your eye. What's that pretty circle of color called? Now, the circle of blackness in the middle is the pupil. Is that a thing? No, it's a hole. Did you know that? You can't pick up a pupil. It's a hole. Why is it black? Because there's no light bulbs in the back of your eyeball. When you take a picture sometimes, and the flash goes in to the hole, it lights up the eyeball, and that's when you get the what? That red eye, pink eye thing in your pictures. Nope, it's not because you're evil. The colored part, you have blue eyes, you have brown eyes, that's called the iris. And that's actually a muscle, a group of muscles going in different directions. And they are going to do what? Control the amount of light that comes in so you can see. The hole gets really small, yes. Next semester when we do special senses, we'll look at some of the different reactions of those muscles in the lab as well. And then they talk about the erector pili muscles on the hair follicles that cause goosebumps. So, skeletal muscle. We look at this diagram here. It should look somewhat familiar to you. What does it kind of look like? Yeah, the nerve you just went through, right? Remember I said we're going to use the same terms, except instead of what? Instead of perineurium, we're going to use the term perimysium. So the gross anatomy of skeletal muscle, you pretty much know it already, just change the back end of the word. Okay? So it's got a nerve supply, it's got a blood supply, and those connective tissue sheaths that cover it are very similar to what we saw in the nerve. So going around the outside, what do we call that? The epimysium. And the epimysium extends out after the muscle and attaches itself to bone to attach muscle. What do we call that extension? It's called a tendon. So a tendon is an extension of the epimysium that attaches muscle to bones. Then, remember my box of straws analogy that we used for the nerve? Okay, same for the muscle. The muscle fiber, and this is the one confusing thing when we read the text and we discuss muscles. When we describe a muscle cell, it is called a muscle fiber. So the term muscle fiber and muscle cell, that means the same thing. Yes? It's important for you to remember that because inside each muscle cell, guess what there is? Fibers. But we don't call them that. What do we call drills? Okay? So, each muscle cell, and this is skeletal muscle, so these cells are quite large. Each muscle cell is multinucleated, has more than one nucleus, and is packed full with myofibrils. Groups of these muscle fibers, remember we're talking about a cell now, are wrapped by membranes called what? Perimysium. And they wrap little bunches called what? Fascicles. Anybody having steak for dinner tonight? There you go. You could dissect it. My family loves when I do that. Oh, look at this. They're like, just eat it, Mom. Jeez. You don't want to know what part it is, just eat it. You don't want to know what it does. I feel like they could learn something at a meal. The meat is tougher because there's less fat. What we're going to find in muscle, especially skeletal muscle, I got a sneeze coming. Excuse me. Muscle, as well as fat, acts as a protection mechanism for different organ systems. And around different portions of muscle groups, we might find more fat than less fat. So when you're picking a cut of meat, a lean cut of meat is what? It is tough because if you don't cook it right. You can't just throw it on the grill and let the flames go with a lean cut of meat because there's not enough fat in it to melt as you cook it and keep it moist. So different muscles in different areas, depending on the animal, depending on the organism, will have more or less fat marbled through it. So when you go to pick your meat again, you've probably heard that. Who's got the best marbling? That tends to be the juiciest steak. And that's basically fat that's running through all of those little fascicle groups. Let's have a barbecue. It could be an anatomy lesson. Works for me. Yeah, dissect it and eat it. All right, so the terms are the same. Endomysium is going to be between each of the individual muscle fibers or muscle cells. Fascicles and perimysium are there as well. So when we look at it microscopically, that little diagram up there, we see this is a cross section looking into the muscle. In lab, we're going to look at two different muscles, two different ways once we hit muscles. But you're going to see that the longitudinal section, looking at these cells coming this way, was something very similar to what you looked at when we did tissues. Remember, it kind of looked like a, I always describe it as telephone poles. All right, come on. Oh, there we go. Huh? No, it's just me. So, the other thing we need to know about muscles is where they attach. And we learned this a little bit already, didn't we? The origin and insertion. We already know what that means, right? So the origin of a muscle is at the what? Immovable joint. For who? Now we have to start learning the names of the muscles. Biceps brachii. Its origin is up here in the shoulder cavity or glenoid cavity, and its insertion is down here. When the muscle contracts, the insertion pulls on the movable bone. Yes? All right. So now we're into looking at the muscle microscopically. Okay? So each cell is packed full of little fibers called what? Myofibrils. And remember, all those other cell parts are there too. So we have the nuclei, and in this particular diagram you can see they're purple. I wish they didn't make them purple because all of the cell membrane proteins are purple. They should have changed the color on the nucleus, but anyway, that's my opinion. They're purple. And they're multinucleated. That's just for skeletal muscle. Smooth muscle is one nucleus like everybody else. Cardiac muscle typically has one nucleus. Every once in a while we'll find a couple with two nuclei. And then we have all the same cell parts. What do they call the membrane that surrounds the muscle cell? It's called the sarcolemma. So what's another name for the sarcolemma? The plasma membrane of a muscle cell. Now the arrangements of some of the different proteins, and this one of the diagrams you should look at to kind of tie everything together is on page 280 in your textbook, figure 9.2. And this is where these diagrams are coming from. So you see at the top that familiar thing you looked at on this diagram in the book when you looked at the longitudinal section of muscle, skeletal muscle cells under the microscope. Does that look familiar? Okay, you see the stripes? What did we say about skeletal muscle? It was striated. Remember we had those terms to describe muscle? We said skeletal muscle is striated and voluntary. We said smooth muscle was not striated and it was involuntary. And cardiac muscle was striated and, well, branching, but it was involuntary. Okay? Now we're going to get to the meat and potatoes of the stripes. Why the stripes? Well, it's because of those little myofibrils that are jam-packed into each of the muscle cells. And each of them are composed of proteins. Groups of proteins all banded together to form these little myofibrils. And the arrangement of those proteins is what's going to give us that striation appearance in the skeletal and cardiac muscle cells. So let's talk a little bit about how they are arranged. First of all, I'll describe the stripes. Because of the arrangement of fibers, myofibrils, we see stripes because there's thicker proteins and thinner proteins that line up very structured in nature. I'm going to skip to the next one and then we'll come back to this one. So who are these proteins I keep talking about? Well, these are regulatory proteins, and you might have heard their names way, way back in chapter, I think it was chapter 3, when we talked about intermediate filaments and microtubules, when we discussed cell parts. Well, these are very similar in structure to those. There's two major proteins that help to make up myofibrils. The one in the middle is this guy here. And these are called thick filaments. It's not just one, it's a whole bunch. Anybody golf? Anybody know what a golf club looks like? Good. Long shaft, little head, right? Huh? Well, if you're a driver, yeah, let's not get into the different clubs. Golf club, right? All right. Now, that fiber is made up of, vision the golf club. If I took a whole bag of golf clubs and I dumped them out and I put a band around them to hold them on, that's what this thick filament looks like. And that's, see all the little golf club heads hanging out there? This is called myosin. So it's like taking 2 bags of golf clubs, pulling them out and sticking them together at their handles to create the thick filament. Can you see that now? Doesn't it look like that? So the other one that makes up this arrangement in the myofibrils is another regulatory protein called actin. And actin kind of looks like a chain of pearls. Okay, little beads. And if we take 2 chains of pearls and kind of twist them like that, we have a thin filament. Again, the thin filament is called actin. And actin and myosin are going to interact together to cause a muscle to shorten or lengthen. The other proteins we're going to see that create this sarcomere are these little things, and they kind of look like bungee cords, don't they? They're called titin. And they're going to help hold all of these guys together. Because they're so highly organized and stacked very uniformly, they create these little units called sarcomeres. So this whole thing up on the board right now is called a sarcomere. And if I take a bazillion sarcomeres and glue them all together, they are going to create not a bazillion, maybe 1,000. We're going to create a myofibril. You with me? I'm going back. You see them? Let me point out one more thing before I go back. The protein that holds together the 2 bags of golf clubs that make up the myosin creates something called the M line. And there's sort of a space, the way the little sarcomere is set up here, that just has thick filament. That's called the H zone. And the Z discs are what delineate the sarcomere. Z disc is also a type of protein that holds everybody in. So from one Z disc to the next Z disc, we have one what? One sarcomere. Oops. All right, so we'll go back to this. This is my Z disc. See it? What is here in this space? Just what? Z disc and who? What's the blue thing? Just the thin filaments, right? The actin. That creates a lighter band. What's it called? The I band. In this area, we have a region that overlaps thick filaments and what? Thin filaments. So it's going to be a little darker, isn't it? What's that called? The A band. Now, right in the middle of the A band, we have a little area that might be a tad bit lighter, and this is a muscle when it's relaxed. And what's that called? It's called the H zone. Okay? So here's another I band on the other side. So you should know this. I know it's a pain, but best diagram for you to look at, page 280 in your textbook. The other thing we're going to find associated with the proteins myosin and actin is two proteins that are going to help regulate muscle contraction. The reason muscles shorten is because of the attraction between the heads on the myosin molecules, and doesn't that look like a golf club? See? Golf club? There's a binding site here for these actin subunits. And when a muscle is relaxed, they can't attach to each other. When a muscle is at rest, the reason they can't attach to each other is because of two other proteins that are very important in the makeup, microscopic makeup of muscle cells. And that is, ready for the names? Tropomyosin, the big long one that covers all those binding sites on the actin molecule. And then the short one is troponin. See, the short word is the smaller protein. The long word is the longer protein. Yes? These two regulatory proteins are going to cover the thin filament and prevent the myosin heads from binding to the actin molecules when a muscle is at rest, when the sarcomeres are nice and wide open. Now, if I take a peek inside a muscle cell, this is how those myofibrils are arranged within the cell. Remember, the cell's got other parts, doesn't it? What other parts does a muscle cell have? Well, I see some really nice looking mitochondria here. That's what you were saying. I know that's what you were thinking. What are those for? Yep, cellular respiration, ATP production. Do we need ATP for muscle contraction? Yes, so we're going to learn that oh so well in a few seconds. The other very important pieces of that muscle cell that are going to help with the whole muscle contraction is these little railways. And in other cells you've already learned about, they were called endoplasmic reticulum. And guess what they're called in the muscle cell? Sarcoplasmic reticulum. Whoa, I know. So you know the word, you just got to change the beginning of it. The other very important part associated with muscle contraction chemistry is not only the sarcoplasmic reticulum, but a certain area of the sarcoplasmic reticulum called the terminal cisternae, which is right near these little tubes that kind of run this way through and around all of those myofibrils. Those little tubes are called T-tubules. And they're kind of like little train stations for ions to run down? The T, that's right. Because I was born in Boston. So, we know what the T is, right? Well, we do. I do. The train. Well, it's a train too. It's up above. Yeah. He knows what he's talking about. No. No, I missed the food there. That's about it. Nothing else. Well, my family, sometimes. So T-tubules and terminal cisternae are all part of very important players in the chemistry of the muscle contraction. So we need to know all of these players. We need to know all of their roles and how they work together for a muscle contraction. And what is a muscle contraction? Well, remember that sarcomere I introduced you to before? Well, we saw the picture. We know all the players, right? That's a muscle at rest. Can you see all the stripes? So when you look at it under the microscope, that's why you see the stripes, because of all the I bands and the A bands and the H zone and the M line and all of that good stuff. Those are stripes. Yes?
Okay. If you look at something under the slide, how can it be contracted if it's not
contracted?
It's not contracted. Well, no, I'm talking about the bottom line.
Well, this is a special microscope that we don't have in our classroom that can look at cells that are alive.
Okay.
Yeah, because yours are... So that's what you see. But this shows us a muscle when it's contracted. And what happens to the sarcomere when a muscle contracts? It's that little unit that we just described from Z disc to Z disc shortens. And how does it do that? Well, that's what we're going to talk about when we talk about the sliding filament theory. So basically, the whole thing shortens. And the reason it shortens is because I'm going to free up those little myosin heads who are so anxious to grab onto actin. I'm going to free up those binding sites. With the help of troponin and tropomyosin, I'm going to free up those binding sites so that myosin can attach to actin and then actually pull it towards the center of a sarcomere. Okay? So this is where the movie comes in. So, again, I usually spend time describing this, and then everybody looks at me like I have 27 heads, and it's not working. So, I decided, and let me go from the beginning because I want you to see how to get here. Oops. Can't see if I push it down to the bottom. Come on. Was there a dance tonight?
Yep. There's a dance tonight.
Okay, you get to interactive physiology through what? Mastering, but you can also get to it this way. Did you know that? interactivephysiology.com, and you put in your login, your regular good old login. I'll put in my login. And once you get to interactive physiology, you're going to see a whole bunch of choices over there. Okay? Does this look familiar to some of you? Hopefully, because what did you use it for? To help you study the nervous system. Some of you like, I didn't do that. Well, you're lost. So, at the top of the list, you have the muscular system. And the one I want to go through with you now is called the sliding filament theory. Okay? So I don't have to talk. Is it too loud?
The current theory of how a muscle cell contracts is the sliding filament theory, which states that the contraction of a muscle cell occurs as the thin filaments slide past the thick filaments. During contraction, the sarcomere shortens and the thin and thick filaments overlap to a greater degree.
In the library, but you want to bring your what? Bring your little headphones because they don't want to hear this nonsense, okay? And that's how you use it.
Your goals for learning are
figure those out on your own.
The sliding filament theory. Your goals for learning are to explore the sliding filament theory of how a skeletal muscle contracts involves the activities of 5 different molecules plus calcium ions. The 5 molecules include myosin, actin, tropomyosin, troponin, and ATP. In addition to these 5 molecules, calcium ions are also involved in the process of muscle contraction.
What can I do? Anything in red you can go click on and it'll define for you. Okay? So that's cool, right? Plus, I didn't quite get that last sentence. What can you do? Play it over again.
Now we will explore how each of these molecules participates in the contraction of a sarcomere. Now we will explore how each of these molecules participates in the contraction of a sarcomere.
The golf club.
Click a thick filament to examine its structure.
Or maybe that one. Or maybe that one. Or maybe...
Thick filament, red filament.
Like the word.
Smarty pants.
The first molecule we will look at is the protein myosin. In skeletal muscle cells, the first molecule we will look at is the protein myosin. In skeletal muscle cells, the myosin molecules are bundled together to form the thick filaments. Click the thick filament to see an individual myosin molecule.
What? Of course it is. Except that one's not working. What's going on? No, you just jinxed me. Oh no, don't do this to me. Okay, wait a minute. Don't do it. Oh man. Come on. Where's the refresh button? Back where? Up here? To the right? Damn it.
The first molecule we will look at is the protein myosin. In skeletal muscle cells, the first molecule we will look at is the protein myosin. In skeletal muscle cells, the myosin molecules are bundled together to form the thick filaments. Click. The shape of an individual myosin molecule is similar to a golf club with 2 heads.
She stole that.
The shape of an individual myosin molecule is similar to a golf club with 2 heads. Myosin has several important functional features. The head, or crossbridge, has the ability to move back and forth. The flexing movement of the myosin head provides the power stroke for muscle contraction.
Use this. And don't think
Click the head of the myosin
silly.
molecule
But when
to
I
see
press
it move.
This, it's going to make a noise. And that stupid little noise is going to make you what? Remember this. And you can keep pressing it and annoy people too. It's actually, my muscles now at this age are actually making that noise when they move. Don't you dare freeze. Come on, baby, you can do it. Oh, I'm getting mad now. It's our crack IT department.
You are
Oh, come on. We already saw that.
The shape of an individual myosin molecule is similar to a The shape of an individual myosin molecule is similar to a Another feature of the myosin molecule is the hinge portion of the linear tail. This allows vertical movement so that the myosin head can bind to actin, the thin filament. Click the tail to see this movement.
Oh, wait, there's more.
The combination of these two hinge points allows for the necessary binding and power strokes of the cross bridges. Click anywhere on the myosin molecule to see both activities.
So that's called the power stroke. That's what happens when myosin reaches up, grabs onto actin, and does what? Pulls towards where? The center, the M line. And what's going to happen to the H zone? It's going to disappear.
The crossbridge has two important binding sites. One site specifically binds ATP. The crossbridge has two important binding sites. One site specifically binds ATP, adenosine triphosphate, a high-energy molecule. Notice the position of the crossbridge. This is called the low-energy conformation. Click the ATP molecule to see it bind to the crossbridge.
The binding of ATP transfers energy to the myosin head as ATP is hydrolyzed into ADP.
The binding of ATP transfers energy to the myosin head as ATP is hydrolyzed into ADP and inorganic phosphate.
The only way that myosin
Click the ATP molecule to transfer energy to myosin.
Is if it binds to ATP. When binding to ATP, it's kind of like changing the shape, because chemistry changes the shape of molecules, right? So when ATP binds to that head, it's kind of like changing the shape of that head and allowing it to go back down to rest. In the meantime, there's chemistry that goes on there. And we're going to break that ATP into what? We're going to break it into ADP and phosphate. So when I break phosphate
now the myosin head is in its high energy conformation.
Well, the muscle's going to relax, but what do I do with that? What have I done now? I changed it. I released what? I released energy. The molecule's going to hold on to that energy, and guess what it's going to use that energy for? The next time it's going to do what? Reach up and grab. And when I reach up and grab, what am I going to do to that myosin head? I'm going to change the shape of it and kick off that ADP and phosphate and wait for another what to come along? ATP. Come on. You can do it.
The second binding site on the myosin head has a strong attraction for binding to actin, as we'll see later in
the second binding site on the myosin head has a strong attraction for binding to actin, as we'll see later in the cross-bridge cycling animation.
Now that we've examined the structure of thick filaments, let's look more closely at thin filaments.
Now that we've examined the structure of thick filaments, let's look more closely at thin filaments. Thin filaments are composed of three molecules, actin, tropomyosin, and troponin. Click a thin filament to examine the arrangement of these three molecules.
The actin portion of the thin filament is composed.
Actin is the major component of the thin filament. The actin portion of the thin filament is composed of actin subunits twisted into a double helical chain. Each actin subunit has a specific binding site to which the myosin head binds.
Fish eggs. That's a good one. It's so imaginative. Oh, come on. 13. I give up too.
The regulatory protein tropomyosin is also part. To expose the binding
The regulatory protein tropomyosin is also part.
molecule must be moved aside.
To expose the binding sites for binding with myosin, the tropomyosin molecule must be moved aside. This is facilitated by the presence of a third molecular complex called troponin. Troponin is attached and spaced periodically along the tropomyosin strand.
They're going to either cover up the actin binding sites or allow them to be exposed so myosin can grab on. So think of troponin as little fists holding on to the rope that is tropomyosin. When troponin comes in contact with an ion called calcium, it's going to change shape. Watch my hands. And pull those ropes, tropomyosin, away from the binding sites. That's going to allow for the two molecules to now bind.
Troponin by itself is not able to move the tropomyosin away from the binding sites. This process
Troponin by itself is not able to move the tropomyosin away from the binding sites. This process requires calcium ions. After an action potential, calcium ions are released from the terminal cisternae and bind to troponin. This causes a conformational change in the tropomyosin-troponin complex, dragging the tropomyosin strands off the binding sites. Click the terminal cisternae to release calcium ions and see this effect.
It's part of what? No, we're inside the muscle cell now. The terminal cisternae is part of the endoplasmic reticulum, remember? All those little canals inside the muscle cell? The terminal cisternae is lined right up with the T tubules, okay? And we're going to talk about the chemical changes that occur there to force these little calcium ions out of the terminal cisternae. So this is what happens when calcium is released into that space. So they change shape and pull those ropes away.
Before seeing how all these components work together in a complete Crossbridge cycle, click the link button.
Before seeing how all these components work together in a complete Crossbridge cycle, click the link buttons to review any of the organic molecules or calcium ions.
Button, it'll take you back where they introduced it the first time.
The 5 organic molecules and the calcium ions function together in a coordinated manner to cause
the 5 organic molecules and the calcium ions function together in a coordinated manner to cause the thin filament to slide past the thick filament. We will first show an animation of a single Crossbridge cycle and then describe this process step by step on the following pages.
So this is what we're going to
see the entire process of Crossbridge cycling, click the terminal cisternae.
So the first thing that happens is a nerve impulse. We're going to talk about that after we go over this as well. That nerve impulse will cause very similar change to what we saw when a nerve caused another nerve to fire off. Remember when the neuron secreted a chemical? What was that chemical called? Acetylcholine is one of them. Neurotransmitter. And then the next cell in line had some sort of receptor for that neurotransmitter. And what happened to the next cell in line? That neurotransmitter typically bound to a sodium-potassium pump, opened up the gates. What rushed in? Sodium rushes in. And that's why when you see me hit this, it's going to say action potential. Because the rushing in of sodium and then taking a ride down the T is what's going to cause the calcium to be forced out of the terminal cisternae. So a nerve impulse is going to stimulate this whole thing to happen. You with me? I understand if you're not with me, because the nice thing is you can go and look at this again. Oh, shoot. Sorry.
As you've just
Shh, shh, shh.
the 5 organic
As you've just the 5 organic
Yes? Yep. So, you are going to have to understand all of the different steps that just happened.
Essay question?
Yeah.
Thank you, Maria.
as you've just seen, Crossbridge cycling.
And this will explain them for
For
you.
purposes of easier understanding,
As you've just seen, crossbridge cycling is a continuous event. For the purposes of
Separate
easier
steps that could be
understanding,
put in
however,
order on
let's
an
break it
exam.
down into 6 separate steps.
2.
1. The influx of calcium triggering the exposure of binding sites on actin. 2. The binding of myosin to actin. 3. The power stroke of the myosin head that causes the sliding of the thin filaments. 4. The binding of ATP to the myosin head, which results in the myosin head disconnecting from actin. 5. The hydrolysis of ATP, which leads to the re-energizing and repositioning of the myosin head. 6. The
convenient
transport of
steps that could
calcium
possibly be
ions
put in
back
order
into
on
the
a
sarcoplasmic reticulum.
exam.
The first step is exposing actin's binding sites. When a muscle cell is stimulated, the action potential
The first step is exposing actin's binding sites. When a muscle cell is stimulated, the action potential brings about the release of calcium ions from the terminal cisternae of the sarcoplasmic reticulum.
change in conformation.
The calcium ions flood into the cytosol and bind to the troponin, causing a change in conformation of the troponin-tropomyosin complex.
Click the terminal cisternae.
This conformational change exposes the binding sites on actin. Click the terminal cisternae to start the process.
That's it. That's for that step.
When a binding site on actin is exposed, an energized myosin head can bind to it, forming a crossbridge.
When a binding site on actin is exposed, an energized myosin head can bind to it, forming a crossbridge. Click the energized crossbridge to begin the binding.
The binding of myosin to actin brings about a change in the conformation of the myosin head, resulting in
The binding of myosin to actin brings about a change in the conformation of the myosin head, resulting in the release of ADP and inorganic phosphate.
of the sarcomer.
At the same time, the myosin head flexes, pulling the thin filament inward toward the center of the sarcomer. This movement
So
is
when they
called
say
the power
conformational
stroke.
change, what's that mean?
Click the energized crossbridge to see this activity.
Changes what? Changes shape. And I use my hand on this one because I think of a hand grabbing onto something, and then when it changes shape, it's gonna let go.
The chemical energy of ATP has been transformed into the mechanical energy of a muscle cell contraction.
The chemical energy of ATP has been transformed into the mechanical energy of a muscle cell contraction.
In order to disconnect the myosin head from actin, an ATP molecule must bind to its site on the myosin
In order to
head.
disconnect the myosin head from actin, an ATP molecule must bind to
So in
its
order to
site on the
change
myosin
shape
head.
that's now
Click the
holding on
ATP
tight
molecule to
of
disconnect
that
the myosin head.
I have to have some sort of chemical reaction to change the chemical configuration or the shape of that molecule. So now it lets go.
The release of the myosin head from actin triggers the hydrolysis of the ATP molecule into ADP.
The release of the myosin head from actin triggers the hydrolysis of the ATP molecule into ADP and inorganic phosphate. Energy is transferred from ATP to the myosin head, which returns to its high-energy conformation. Click the bound ATP to trigger
Now, you know why they call
hydrolysis.
that the high-energy configuration? Because the energy released from the hydrolysis of ATP is being held in the molecule until it does what? Reaches up, grabs on, and power stroke happens again. Changes shape.
In the final step, calcium is actively transported from the cytosol back into the sarcoplasmic reticulum.
In the final step, calcium is actively transported from the cytosol back into the sarcoplasmic reticulum by ion pumps. As the calcium is removed, the troponin, tropomyosin complex again covers the binding sites on actin. Click a calcium ion to see this process.
The active transport of calcium involves specialized ion pumps in the membrane of the sarcoplasmic.
The active transport of calcium involves specialized ion pumps in the membrane of the sarcoplasmic reticulum. These pumps must be energized by
stopped the nerve
ATP.
impulse from taking
Click the
place?
calcium ion pump to see
Once we
active
went through this whole
transport.
action potential business, what stopped the neuron from firing off? Huh? Not reaching threshold because of what? The ion pumps, right? The ion pumps turn back on after we degrade the neurotransmitter, right? Same thing's going to happen here. Those little pumps are going to become paralyzed. And they're going to become paralyzed when sodium rushes in and runs down the T. They're going to kick back into gear now and do what to calcium? All that calcium that got let out, what are they going to do now? Suck it back into the terminal cisternae. Oh, don't freeze. Come on. Click the pump? Okay. So not only do we need ATP for this, but we need it for a muscle to relax. Yes? See, I got this down.
To see the entire process of crossbridge cycling, click the terminal cisternae.
So now all sticks
To see the
together
entire
again.
process
See
of
it
crossbridge
again.
cycling, click the terminal cisternae.
Now, does this happen just like once? No, in order for it to shorten, guess what's going to happen?
In contracting muscle, we see multiple crossbridge cycles. Here are 4 myosin heads.
In contracting muscle, we see multiple crossbridge cycles. Here are 4 myosin heads which cycle at the same time. Note that during a contraction, all myosin heads would cycle, but not all are bound or
That
disconnected
happened, if
at the
everything
same
bound and let
time.
go at the same
Click
time,
the terminal cisternae
would
to
you have a
begin the
nice
process.
even shortening? No, it would be like playing tug of war, 2 teams, and the whole entire team pulling and letting go at the same time. Would that work?
That's funny.
No, it would be funny. It would be a good funniest home video, I think, but it wouldn't work. All right, don't get carried away.
In this view, several myosin and actin filaments are interacting to demonstrate the sliding filament.
In this view, several myosin and actin filaments are interacting to demonstrate the sliding filament theory of muscle contraction. Notice that although the sarcomere shortens, the length of each myofilament does not change. However,
So remember, this
watch
is the
as the width
sarcomere
of
at
the
rest.
H zone
These are the
changes.
stripes you
Click a
see,
thin
all of
filament
that
to
good
start the
stuff, right?
contraction.
This is the H zone. This is going to shorten, but these fibers aren't going to shorten, are they? No, the whole unit is just going to pull together closer. That's what they say when they say the fibers don't change size. What am I clicking? See? One more time. So see all those little heads? Yeah, exactly. It's like a tug of war. That's a muscle contraction. I know. Don't do that. It's not good for the machine. All right?
We've seen that ATP plays a key role in the contraction of muscle. Before we study muscle metabolism,
We've seen that ATP plays a key role in the contraction of muscle. Before we study muscle metabolism, let's review ATP's role in 1.
The other thing about this program,
here's a summary of what we've covered.
it summarizes.
The sequence of events.
And so you see the button I just clicked?
Here's a summary of what we've covered.
Over here in the corner.
The sequence of
Quiz.
events.
So now you can go do what? See if you know what the heck they're talking about. And again, the cute little noises will come up. So it says, click and hold on the arrow next to each participant in the sliding filament theory and choose the correct name. What is that? It's myosin. I thought I did too. I wanted to hear the bad answer. Why is it doing that? Okay, hold. Okay, I gotta hold. What is that? Oh, wait a minute. I'll say ATP. So, see? I know. See the answer?
Well, we already heard it.
I know. All right. You gotta hold it anyway. There you go. Okay? So you can do little quizzes. And I want to go through the quiz, so hopefully I'll see the end. Oh, sometimes it'll go, yay! And throw like confetti. Okay, so you can quiz yourself. Good thing to do, right? All right, so that is the sliding filament theory. Should you use that? Yes. All right, you just be quiet. Yeah. All right, so back to the PowerPoint. What? All right, so outline for you here is what? What is this? In this diagram. No, this is the lead up, okay? This is the lead up in phase 1 to what we just saw in the sliding filament theory, the contraction part, right? What causes all of this change to take place? Yeah, an action potential, a nerve impulse. And that's what they start to describe under nerve stimulus and events at the neuromuscular junction. What the heck does that mean? Where the neuron, those axon end bulbs, meets the what? The muscle cells, or muscle fiber, or the sarco, what do they call that? Sarco something that wraps around the cells. The sarcolemma that wraps around the cells. So on page 286 and 287 in your textbook, we see the events at the neuromuscular junction. So we'll pick up with that on Tuesday of next week. Don't forget to sign the what's this. Is that what you're asking for? No. Don't forget that next week, all you're going to see me on is Tuesday. I know it's upsetting. And what else did I was going to tell you not to forget? Oh yeah, don't forget to read this chapter and look at your interactive physiology. Who's got the sign-in sheet?